My name is Jakob. I'm an assistant professor at the section for cell biology and physiology at the Department of Biology at the University of Copenhagen, and I will be chairing the coming session. I think the organizers have put together a really nice program for us, focusing on the intracellular mechanisms that control glucagon secretion from alpha cells. And I have been given the daunting task to try and give you a brief introduction to glucagon secretion and alpha cell function. And so when I got this task, I thought, well, how do I do that? So I'm going to give you the briefest of introductions, just a couple of slides on how glucagon secretion is regulated, and then I'm going to give you some thoughts on what I like to study the most, namely alpha cell metabolism. So you probably know that we are looking at an islet here, and we have glucagon in green and insulin in red, and as you can see, a very nice staining. We know that the alpha cell sits in these microorgans in the pancreas called islets of Langerhans, and they are highly heterogeneous in their cellular composition. We have beta cells that produce insulin, we have delta cells that produce somatostatin, and we have alpha cells that produce glucagon. And we think that alpha cells depend very much, of course, on glucose, but also on the cells in the surrounding environment. We therefore, in the field, currently think that glucagon secretion is regulated in a multimodal fashion. So we have two modes. One is an intrinsic effect, whereby glucose directly regulates glucagon secretion by acting on the alpha cell. And the other is a paracrine effect, where for instance, secretion from beta cells by insulin secretion or somatostatin secretion from delta cells leads to paracrine inhibition. As you can see, the intrinsic effect is mostly active at lower glucose levels, and at higher glucose levels, we think the paracrine effect takes over. And this leads to this combined effect that you see here, that we all know and love, the inhibition of glucagon secretion by glucose. The prevailing hypothesis of how the paracrine control of glucagon secretion works looks something like this. This is one way of looking at it. I think we're going to hear today that there are probably modifications to this scenario, but we think that the beta cell secretes insulin, GABA, and probably serotonin to inhibit the alpha cell. The delta cell signals by secreting somatostatin, and then the alpha cell can regulate glucagon secretion in a feed-forward mechanism by secreting glucagon and glutamate. And the intracellular scenario looks something like this. Not exactly simple, but I will try to sort of, can I have a laser pointer here? I'll try to explain. Well, you can see there are stimulatory hormones up here, adrenaline, vasopressin, GIP, and glucagon. They stimulate through G-coupled protein receptors to elevate cyclic AMP and activate PKA, and possibly also EPAC2. And this leads to activation of calcium channels in the membrane, effects directly on exocytosis of the granules, and also effects on ER calcium that all leads to an elevated release of glucagon. So we think that that cyclic AMP plays a central role. Also for the inhibition, of course, we have somatostatin and insulin as the major inhibitory paracrine regulators, but serotonin also seems to play a role. And especially somatostatin and serotonin is thought to regulate glucagon secretion through by decreasing cyclic AMP, but also by regulating electrical activity directly at the membrane through, for instance, GERK channels, or by activating indirectly the GABA receptor, so GABA can signal to inhibit glucagon secretion. So this may seem relatively simple, and I think any cell biologist would say, well, there must be much more to this, because we also know that PKA and AKT play other roles in cell biology than just what is shown here. So I think there's some interesting exploration in store for us in the future. If we look at the prevailing hypothesis of that regards the intrinsic control of glucagon secretion. We mainly view alpha cells as consumers of glucose. And the idea is that at higher glucose levels, extracellular glucose concentrations increase. This is metabolized in the mitochondria and oxidized to produce an elevation of ATP. This changes the relationship between ATP and ADP in the cytosol. And here we have disparity in what we think is happening. There are two main hypotheses suggested. One suggests that we activate the circuit pump, thereby dissociating, increasing intra-ER calcium. And this dissociates channels on the ER with proteins on the ER with channels, store-operated channels at the membrane, and this changes calcium influx into the cell. The other hypothesis is based on sensing of the ATP-ADP ratio by the ATP-sensitive potassium channel, and that changes membrane potential to something something like what you see here. And don't be afraid, this is just membrane potential over time. You see action potentials firing as the vertical lines, and down here you see that here you see the basal membrane potential, and you can see we depolarize this membrane slightly when we add glucose due to the increase in ATP, and we get shorter action potential heights. And this also leads to reduced calcium activity, as you can see here, where we used an alpha cell specific GCAMP to measure calcium oscillations in response to glucose. So this is all well and good, but alpha cells function when there's not much glucose around. So I think that's an interesting point that maybe we should think a little bit about. Well, alpha cells are active, for instance, when we exercise or when we're fasting. Here's just young men exercising for 3.5 hours until exhaustion at 55% of their VO2 max. And that is really tough when you are exercising that long. And you can see they have a beautiful drop in plasma glucose levels, and they have an increase in non-esterified fatty acids, and then we have a beautiful increase in glucagon. And one could question whether glucose could provide the energy needed to drive glucagon secretion in a situation like this. And we don't think so, but we think there might be other substrates. And the clue really came from a paper published in 2013 by Patrick Roseman's group, where they show that if you treat islets with oligomycin, you see an inversion of the glucagon response to glucose. So in black you see here what I also showed you before, that when we add glucose, we see a decrease in glucagon secretion. But when we start to inhibit ATP, something really interesting happens. We see elevation in glucagon secretion with 6 millimolar glucose, but glucagon secretion at low glucose seems to be inhibited. And this could suggest that there might be other substrates that are important. My favorite suggestion is fatty acids, and this has been something that has been debated in the literature for a very long time. I think the first data on whether fatty acids regulate glucagon secretion came out in 1967, and at that point no one really agreed on what was happening. But one of the really interesting findings, I think, is what you see here. Here they have treated volunteers with intralipid and heparin, and what you see is you get a massive increase in free fatty acids, circulating free fatty acids, and this of course leads to an increase in immunoreactive glucagon. So this suggested that glucagon could be stimulated by fatty acids. We can also show this in mouse islets. This is data from Patrick Roseman's group again, where they stimulated isolated islets with 1 millimolar palmitate, and it increases glucagon secretion at all glucose concentrations. So we thought that was interesting, and then we went and knocked out fatty acid oxidation, specifically at islets, and this is a long-chain fatty acid oxidation, so we knocked out carnitine palmitoyl transferase 1, which sits in the mitochondrial membrane and conveys the uptake of fatty acids into the mitochondria. So when that enzyme is removed, we have no fatty acid oxidation in the mitochondria, and we see lower glucagon secretion at low glucose. So this could suggest that the alpha cells use fatty acids to drive glucagon secretion, at least at low glucose. But I think there's still lots of open questions. What is the mechanism, and do fatty acids stimulate glucagon secretion at physiological concentrations? And now you're probably thinking, hey, wait, Jens said something about fatty acids, but he also said something about amino acids. And I think that's also a really interesting candidate here, because amino acids can stimulate glucagon secretion, as you see here. This is both old data in dogs, but also newer data in mice. And what you see here on the other side is the metabolism of all the amino acids. And you can see they can definitely contribute to the TCA cycle. But whether they do that in alpha cells, we don't know. We don't even know the mechanism by which they stimulate glucagon secretion. But I hope today we will perhaps get a better idea about this. So my humble conclusions are glucose regulates glucagon secretion both through paracrine and intrinsic mechanisms. Paracrine mechanism seems to be driven through cyclic AMP or direct effects on ion channels. The intrinsic effects require glucose uptake, but alpha cells also thrive on non-carbohydrate substrates, and I think this may require that we reconsider the mechanisms that regulate glucagon secretion and alpha cell function. So I hope that this is a lead up to a very good session, and I think we, unless you have some burning questions, then I think we should move on to the next speaker.